Good day, everybody. Today I'm going to be lecturing on the molecules of life. That's why I've titled this presentation, Lively Molecules. In particular, we're going to be focusing on three groups of compounds: the carbohydrates, the lipids, and the proteins. Now, the main thing to remember so far is just that every one of these compounds has the element carbon making them up. So carbon is probably the most abundant element in the entire human body. By talking about the carbohydrates. Now, carbohydrates can be divided into three subclasses: the monosaccharides, the disaccharides, and the polysaccharides. In this slide, we're going to be looking at three of the main monosaccharides. The main one is glucose. Glucose is the primary fuel for the cells. As you're going to learn when we study the nervous system, the brain cannot operate without glucose. And in cell respiration, it's glucose that is broken down to supply ATP for all of our chemical processes. A close cousin to glucose is a monosaccharide called galactose. Now, notice that the structure of glucose and galactose are very similar. Only difference being the placement of the carbon and the hydroxyl groups. That position being like a switch, like right here. On glucose, in this model here, the hydrogen is in this position, but in galactose, it's down here. And hydroxyl group here on galactose, and here on glucose. Now, a third monosaccharide you need to know is called fructose. Now, the thing that all these monosaccharides have in common is that they are 6 carbon compounds. Now, the way these diagrams work, every point, like right here and all the way around, is a point where there's a carbon atom. So in this one, there are 6 carbon atoms, just like there are in galactose. But you look down here in fructose, you'll have 1 carbon atom here, 2, 3, 4, 5, and 6. So all three of these molecules have the same chemical formula, which is C6H12O6. All monosaccharides will have the same ratio of carbon to hydrogen to oxygen, and that ratio is always going to be 1 to 2 to 1. Now, because glucose is the main fuel for the cells, galactose and fructose will be converted into glucose for most cell uses. And that conversion usually occurs in the liver and small intestines. Oh, I'm sorry, not small intestines. Basically, in the liver. Now, the second group of carbohydrates are the disaccharides. The prefix di means 2, and saccharides are sugars. So the disaccharides are the double sugars. Now, disaccharides are formed by combining 2 monosaccharides through a process called dehydration synthesis. Dehydration synthesis is the removal of water to form a chemical bond. Take, for example, the molecule sucrose, which is normal table sugar. If you look at the diagram here, sucrose is composed of a molecule of glucose and fructose. When they combine chemically, the hydroxyl group of the glucose combines with strips of hydrogen away from the hydroxyl group of fructose, and H2O is squeezed out, and a chemical bond is formed. Sucrose is your normal table sugar. Now, maltose, or malt sugar, is formed the same way. Again, it's dehydration synthesis, but now maltose is a different combination of monosaccharides. It's basically 2 glucose molecules combining together to form maltose. Well, I don't have a diagram of this showing the same process, but lactose, which is milk sugar, forms the same way. Only lactose is going to be a combination of galactose and glucose, and you'll have the same process of dehydration synthesis. Now, to split these molecules apart, you need an opposite reaction. To split those molecules apart, you have a process called hydrolysis. And again, if you break the word down, hydro means water and lysis means to split. So if you look at the diagram down here of a polymer, a 4-chain polymer, water is added at this bond here and it splits it apart into hydroxyl and hydrogen, and the molecule is split apart. Now the third subgroup of carbohydrates are the polysaccharides. Prefix poly means many sugars. So what you've got here are long chains of glucose. When you put long chains of a molecule together of the same repeating subunits, in this case glucose, you get what we call a polymer. Now, in polysaccharides, in the human body, we need to concern with ourselves here, there's two major polysaccharides, glycogen and glucose. Glycogen is the storage form for glucose that's stored inside our muscles and our liver, while starch is the way the long chains are stored in a plant. So our bodies are concerned with the starch when it comes to breakdown in our digestive system, and glycogen is a storage form inside our liver. This is our storage form for glucose so that we can fast during the night. The only difference between these two is the degree of branching. Starch are pretty much straight chains. Glycogen is many chains, or many branches. Now the functions of polysaccharides fall in line with all carbohydrates. They're a main source of fuel, as you see with glucose. But again, glycogen is just long chains of glucose, so we can sleep at night. And of course, structural components. You will find both glycolipids and glycoproteins making up parts of the cell membrane. This is going to be used mostly for cell identification and cell recognition. The second group of organic molecules that I wanted to discuss with you today are the lipids. Now, lipids are characterized as being hydrophobic, which means water-fearing. Just like the carbohydrates, they are composed of carbon, hydrogen, and oxygen. However, the ratio is different. As you saw with the carbohydrates, the ratio of carbon, hydrogen, to oxygen is always a 1 to 2 to 1 ratio. In the lipids, there's a much higher ratio of hydrogen to oxygen. You don't see that even 2 to 1 ratio between the hydrogen and the oxygen. That makes lipids much more oxidized than carbohydrates. So you can actually store more calories per gram in a lipid than you can a carbohydrate. So that makes them a very good source for stored energy. And these can be broken down into 5 subclasses as well. Like the carbohydrates can be broken down into 3, lipids can be broken down into 5. Now, one of the subclasses of lipids are the fatty acids, like this picture here. Fatty acids are long chains of carbon atoms bonded together, and they can be anywhere between 4 and 24 carbon atoms long, and they can either be saturated or unsaturated. The picture you're looking at right now is an example of a saturated fatty acid. It's saturated because the carbon atoms are completely filled up with hydrogens. All bonds are completely filled. Hydrogen is occupying all spots here. If there were a double bond in there, then it would be unsaturated. So while this one is saturated, if you can imagine hydrogen atoms missing in 2 spots, like say for example, this hydrogen atom and this hydrogen atom were missing, and the carbon atoms formed a double bond here, then you'd have an unsaturated fatty acid. Now if you have many double bonds throughout the chain, then it's polyunsaturated. Now in our bodies, we have something called essential fatty acids. These are fatty acids that we have to consume in our diets. We do not make these in our bodies. Most other fatty acids can be formed from other molecules, but there are several essential fatty acids that we must have in our diets, usually coming from plants. Now, a second subgroup here are the triglycerides, like you see in this picture here. A triglyceride is a combination of 3 fatty acids and 1 glycerol molecule. In my picture here, this gray shaded area here shows the glycerol molecule, and the brown shaded areas are showing the 3 fatty acids. And just like the carbohydrates, they are formed by dehydration synthesis, squeezing out a water molecule between each one. And just like the carbohydrates, they are broken down by hydrolysis. Now, triglycerides can be classified as either oils, and the classification here is based on their state at room temperature. Oils are basically going to be liquid at room temperature. So we're looking like all plant oils. But fats are going to be solid at room temperature. So this would be like lard and any animal fat. Now, why are oils a liquid at room temperature? Oils have unsaturated fatty acids. So they will form double bonds between the carbon atoms, and that tends to make them stay liquid at room temperature. The next subgroup of lipids, special group of the cells, are the phospholipids. Here, a phospholipid is kind of like a triglyceride, but in its place, a phosphate group replaces one of the fatty acids. So in our picture over here, here's our phosphate group. This is normally where you would have one of the fatty acid chains, and here are the other two. So it gives it a special appearance. And like I said, these do form part of the cell membrane. Another subgroup are the steroids. Steroids are basically carbon chains that, instead of being straight chains here, are into rings. And cholesterol is a good example of this. This is a molecule of cholesterol. Cholesterol is necessary for production of vitamin D, some steroid hormones, and the bile salts. Now, we hear about good and bad cholesterol, and the difference is this. Good cholesterol and bad cholesterol refers to HDL or ADL. HDL is the good cholesterol. That stands for high density lipoproteins. So it has a lower ratio of lipids to proteins. So basically look at it as having less fat, more protein. Whereas the bad cholesterol, the LDLs, have low density lipoproteins, so you have more fat than you actually have protein making it up. Now, the last group of organic molecules we're going to look at here are the proteins. Proteins make up 10 to 30% of the cell mass, so they're pretty important. They are polymers, so they're made of repeating subunits called amino acids. In my picture right here, I've shown you a whole chain of amino acids. So this is a polymer of amino acids or a protein. And this is a picture here of the first level of organization of a protein, simply the sequence of amino acids. Now, the amino acids are composed of a special type of bond, which you see only in proteins, called a peptide bond. It's basically formed by dehydration synthesis, like we've seen before. And proteins can be either classified as fibrous proteins, and some textbooks refer to them as structural proteins. Fibrous proteins are going to be insoluble in water. So they actually support structures. They're very stable, and a good example you've already been exposed to is collagen. Globular proteins, or the functional proteins, are water soluble, and they'll include things like antibodies, hormones, and enzymes. Now, the enzyme's structure determines its function. So anything that disrupts that structure, or how that protein is put together, is going to affect its action. And when a protein loses its shape and becomes unfunctional, we call that denaturation. In my picture here, there is a picture of a protein with a specific shape. Something can happen, like excess of heat, excess salt, anything can disrupt these bonds between the different parts of the protein and cause it to lose its shape. When it loses its shape, it loses its function.